You can see our topic is about designing vaccines based on calculations of the relations of the genetic sequence. 由基因序列的亲疏设计流感疫苗的探讨。In our literature, we are trying to use the mathematical science methods, trying to compare a known virus and the known viruses, and we can compare the sequences of the flu sequences between each others, and we can find out the immunity point. And the immunity point might be the point that can help the scientists to invent the new vaccines. And here we try to find out the sequences of the PP2, HA, and NA. And the reason we want to find about HA is that because you see in this movie that HA is like a key that helps the virus to connect the surface of the cell to get it into the cell and produce a lot of viruses. And the NA here it says that when the new viruses were produced, they are trying to get it out of the cell. And the NA will be like a scissors you see in here. That it will cut it off, and it can get it out from the cell and go and keep on going to affect other viruses.
And in our research, we propose mathematical and bioinformatic methods trying to locate the vaccine immunity points. And in our research, we use two different kinds of mathematical methods. One is UPGNA unweighted pair good method using arithmetic average, and another one is MLE, maximum likelihood estimator. UPGNA is to using a similar matrix to construct the phylogenetic tree diagram. And through each pairing, we combine the most close two biological groups into a high-level group. And in our research, we use the UPGNA to calculate because it is the most frequently used phylogenetic analysis measure. And here is the maximum likelihood function. And in the functions, x1 to xn means the 14 influenza A virus sequences we find out from the NCBI online database. And the theta means the sequence we need to calculate with. And in our research, we calculate with the PP2, HA, and NA sequences. And after we calculate with the probability mass functions, we can find out the relationship between 14 influenza. And here is maximum likelihood estimation of biological sequences. And here we use the two-sign ratio approach to find out the best estimation of the theta. And after we calculate with the two different kinds of mathematical methods, we can come up with the duty tree and ruler tree like these figures.
And then we will find out the 14 viruses on NCBI database, the National Center for Biotechnology Information. We will compare its PP2, HA, and NA. In this ruler tree, it's comparing the PP2, the tree. As this picture shows, you can see that H5 and 3 and H7 and 3 has a close relationship, and H7 and 7, H7 and 9 has a close relationship. These two pictures is before all sequence comparison. These two pictures is after all sequence comparison. Those two pictures is using the MLE, maximum likelihood estimator.
Figure 不是 picture.
Oh, okay, figure. After deleting the head and tail. In this figure, these two has a close relationship, but after all sequence comparison, we can see that the H7 group get closer, and it means that it really has a close relationship. In this maximum likelihood estimator, if we want to have more accurate experiments, we have to delete the head and tail. The same in this picture, we can see that H7 group is just getting closer. And in this picture, we using maximum likelihood. We cut the head and cut the tail, and we just got the middle part because the length of the 14 viruses are the same. Some are short and some are long. So we cut it to yes, and take H5 group for example. The star means three sequences are the same. The one without the star means that it might be red or just different. It may come out with the 17, 20, 23. 70, 20, 23, and so on, and come up with in total 400 different locations. And in traditional invention new vaccine, scientists should do all the sequence by 1,849 experiments. And now, using our experiment, a mathematical method, we just have to do the 400 different locations, and the same in the H7 group. 学长换你。
And all above is about the HA analysis. And I'll keep on continuing to talk about the NA analysis. In this figure, you can see that these two are the before all sequence comparison, and these two are the after all sequence comparison. And these two are the maximum likelihood rooted tree diagrams of the 14 viruses after all sequence comparisons. We can first see here that H7N9 and H10N7 and H7N7's NA sequences has a very close relationship. And H7N3 and H5N3's NA sequences has a really close relationship. But we find out that when after all sequence comparison, we found out that these two didn't have a lot of difference. So it means that the N7 groups and the N3 groups have a really close relationship. And we do the maximum likelihood to get a secondary analysis to get a more accurate experiment. So we can find out that the saying that H7N9 and H10N7 and 7N7's NA sequences has a really close relationship, and the same to the H7N3 and H5N3's NA sequences. So we do the same methods with the above HA, compare it with the sequences, and we can come up with that there are 191 different locations between H5N3 and H7N3, and all of the total length is 1,410 BP. And here the same, the N7 group, we find out 623 different locations, and the total length is 1,438 BPs. So we can see that we can save a lot of time and money by getting the immunity points to invent the new vaccines. So while developing vaccines with biomedical information research, a highly inconsistent ratio is frequently encountered, namely the best location of immunity. So in this research, we are the first one to conduct computer analysis on a known virus, and then compare with sequence of known viruses and vaccines through needle-launch dynamic programming algorithm. So I will now talk about the figure 29, the reverse genetic procedures of vaccines production revealed by this research. So without this mathematical method, we can find out that it is a conventional R&D process for the scientists to invent the new vaccines. And the scientists need to foster new influenza A viruses and run a 2D enterophoresis and keep on this step on and on to find out the parts that the immunity point do, and it can you can get it to invent the new vaccines, and they can do secondary analysis to help the human against the influenza A viruses. So in order to save the time, we add a mathematical science step in here. We call the reverse genetic procedures. We can see that this is a non-influenza A viruses, and this is a standard viruses. We will choose the unknown influenza viruses HA and NA sequences, and to combine together with the standard viruses other 6 sequences in here without HA and NA, the other 6 parts, and we will combine together to form a new viruses, and we can use the new viruses to invent the new vaccines, and the vaccines will have can against the HA and NA of the unknown flu viruses. So with this kind of mathematical science methods, briefly to say, we just need to find out the unknown viruses sequences and compare with the known virus sequences. Just compare the difference between these two kind of viruses. The different point might be the immunity point for the scientists to invent the new vaccines more quickly. So we think that this kind of methods can benefit our humans a lot and save a lot of time inventing a new vaccine. Thank you for your listening.
9分55。